Excitation contraction coupling in skeletal muscle. Now coming to the properties of skeletal muscle, the two basic properties remains excitability and contractility. Here you see the graph. The graph is strength duration curve, and the point B is showing the chronaxy, that is for muscle, that is the excitability of the muscle is less as compared to the nerve, as we had already studied earlier. Now coming to the properties of the muscle, the first property is a property of muscle excitation. Excitation, the muscle action potential is similar to that of the nerve action potential. Some of the differences will be highlighted later. The resting membrane potential is minus 90 millivolt, the action potential being 2 to 4 millisecond, whereas in nerve it was just 0.4 to 2 meters per second. The conduction velocity is very low, that is 3 to 5 meters per second, whereas in a nerve it was variable. It was classified in later classification, and according to the diameter, the conduction velocity was variable. The absolute refractive period here is 1 to 3 meters per second, whereas in the nerve it was very less, it was 0.2 to 2 milliseconds. The excitability, therefore, because of all these features, the excitability is less than that compared to nerve, and the chronaxy is for a longer period. So when we talk about this muscle twitch, a single action potential causing a brief contraction followed by relaxation is known by the name of muscle twitch. It is, as you can see in this graph, it is not starting immediately, but there is a latent period of approximately 2 meters per second. After the start of depolarization, there is a contraction of the muscle in this graph. You can see for the fast muscle fibers, it is 7.5 meters per second, and for the slow it is 100 meters per second. So it is variable depending upon the muscle type. The relaxation phase, you can see in the graph, is more as compared to the phase of contraction. So when we talk about this excitation contraction coupling, there is a process by which a muscle is excited, and that is coupled to the process of muscle contraction. So both of these processes are coupled together or linked together by which a muscle contraction can occur. So they are the sequence of events by which an action potential in the plasma membrane is leading to the cross bridge activity. So let us study that in detail. First question remains, how do a muscle contract? So whenever a muscle contraction has to begin, and it is a voluntary muscle contraction, it will start and initiate in the motor areas of the cerebral cortex. Then the tracts arising from there, corticospinal tract, will end up in the anterior horn of the spinal cord. From there, the neurons will arise, which are known by the name of motor neurons, and they will innervate a particular muscular area which is responsible for the muscle contraction. It will form the neuromuscular junction there. There will be the series of events in the neuromuscular junction resulting in generation of motor and plate potential, then action potential, and then finally we have the excitation process, which is coupled to the contraction process, cross bridge cycling. Finally, muscle contraction will be there, which is followed by a phase of muscle relaxation. So what is this motor unit? Whenever the tracts are terminating in the ventral horn, from the ventral horn there are neurons which then supply the muscle fibers. So this collection of muscle fiber which are innervated by a single motor neuron, they form the motor unit. So when this neuron is stimulated, all the muscle fibers which are supplied by that neuron will contract together. So you can see two types of motor unit, one in green and one in blue. So one type of neuron is innervating all the muscles that are in blue. So this is one motor unit and it is a larger motor unit. And the second one is innervating only the one muscles with green color. So this is a smaller motor unit. So some may be fast or some may be slow. The small motor unit offers greater precision. They are rapidly reacting. Why? Because the fibers are less than 10 fibers per unit. But when we talk of a larger unit, maybe thousands of fibers are there, they are responsible for the coarse movement, or they are slowly reacting. For example, your quadriceps muscles. So each has a different functional role. However, they overlap and then they provide for the coordination of this movement. And when we talk about any muscle, we have the mixed motor units. It can be large motor units, it can be small motor units, it can be variable, it can be few motor units, or it can range from hundreds. So for a weaker contraction of the whole muscles, only few of the motor units are activated, and this we'll discuss later when we discuss the size principle. So when there is an excitation of the motor neuron, it will terminate on the muscle end in the center of the muscle in the form of this neuromuscular junction at nerve and muscle end. So what are the events that occur in a neuromuscular junction? There is a reaching of action potential to the neuromuscular end, opening of voltage gated channel, resulting in mobilization of these vesicles which contain acetylcholine to the motor end, that is to the muscular end, or the area which is present between the nerve and the muscle. Release of this acetylcholine will then act as a ligand to the sodium channels which are present there. Two molecules of acetylcholine will bind with the alpha subunit of this nicotinic receptor, which is NM receptor, resulting in opening of the channel, and we have influx of the ion which is present in high concentration there, and it is sodium ion. So there is a net movement of sodium inside the muscle and helping to generate the motor end plate potential, or the miniature end plate potential. So this will then result in next step. As you can see, step number 4 is the sodium entry, and step number 5 is the local current that has been generated. This local current will reach then a level of threshold, and it will be muscle action potential that can then spread along the muscle membrane. You can see here it is traveling along the muscle membrane. However, we have the muscles, that is the cross bridge formation is in the center of the muscle, or the smooth endoplasmic reticulum with calcium store are present in the inside of the muscle. So this action potential traveling through the membrane will not suffice, as a result of which depolarization of the muscle membrane also occurs through the T-tubule and reaches to the cell interior. And here we have a receptors which are dihydropyridine receptor, which are nothing but voltage gated calcium channels. So once these receptors have been activated, they are mechanically coupled to the ryanodine receptor, which are present in sarcoplasmic reticulum. So the activation of this dihydropyridine receptor, you can see that are in red color, that will lead to the mechanical pulling of the ryanodine receptor, and when they are activated, the ryanodine receptor, there is release of calcium from the sarcoplasmic reticulum because it is high in level. So diffusion of calcium will occur from sarcoplasmic reticulum to the sarcoplasm. As a result of this, the calcium will be more in sarcoplasm, that is, it will reach a level of 10-4 moles from compared to its previous level of 10-7. So what is the role of this calcium? Will then go and bind to troponin C. The troponin C will then cause the binding of 4 calcium ion to troponin C will result in the movement of tropomyosin. The tropomyosin will move into the groove and it will expose all those 7 sites which are meant for interaction with the myosin molecule. So when calcium is present, there is uncovering of the 7 myosin binding sites for the myosin. So actin-myosin interaction can occur. So this actin-myosin interaction occurs by sliding filament theory, also known by the name of walk-along theory, was proposed by Huxley and Huxley in 1954. They proposed that sliding of filament is brought about by a repeated cycle of cross-bridge formation between the head of myosin and the actin molecule. So let us study this step by step. The first step is ATP binding, in which a fresh molecule of ATP, you can see here, binds to the myosin head. The myosin ATP complex, since it has a low affinity for the actin, so there will be detachment of the myosin complex from the actin molecule, and the muscle, if it remains in this form, it is relaxed. So the next step will be step of ATP hydrolysis. The ATP which is present there will be hydrolyzed to ADP and phosphate. The product so formed will be retained in the active site of the head of the myosin. So this complex, that is ADP myosin complex, it is a high affinity for the actin molecule, and there will be cocking up of the myosin head or neck, as you can say, resulting in movement of the head 11 nanometer along the actin filament, and you can see it has moved 2 step or 2 monomer farther along the actin. The next step is formation of a weak cross bridge because of binding of myosin ADP complex with the actin molecule, and you can see it is just 2 monomer away. The next step, release of the phosphate will be there. So myosin is left only with the ATP, and this complex has high affinity for the actin. So there is a transition from the weak cross bridge formation to strong cross bridge formation, and actually this is the rate limiting step that will determine the rate of cross bridge cycling. The next step, or the step here, is the power stroke generation. The myosin neck will now rotate around the myosin head, which is attached to the actin, and this will result in a mechanical force or a power stroke that will pull the actin 11 nanometer distance. So the Z lines, if we talk microscopically, they will come closer, the muscle will contract, and the myosin head will remain still in a position of 45 degree, but it has now stopped sliding. Step 6 is the ADP release. The ectomyosin complex is left in this state, and it is an attached state, a rigid state, and actin myosin head and neck remain in same position until unless the next ATP molecule will come and will bind with this complex. So again, a fresh molecule will be needed. The same steps will be repeated again. So the essentials which are required for muscle contraction, one is the calcium ion. An increased level is required in the sarcoplasm of calcium from its resting state to its active state. Then we require source of energy in the form of ATP, and for each round of cross bridge, approximately 1 ATP molecule is consumed. So the termination will occur when this calcium is pumped back into the sarcoplasmic reticulum or to the exterior. So it is done by two mechanisms. One is sodium calcium exchanger, and the other mechanism being calcium ATPase. So the relaxation will occur. The relaxation phase is slower because the pumps are slower. So the relaxation is prolonged. So the question remains, why actin molecules do not slip back to the resting position in between the strokes? It is because some of the cross bridge are attached while some are returning to the original formation. So basically the cross bridges, some are in hold on position and others are in let go position, resulting in not slipping back of the actin filament. So walk along theory. So this is the basic walk along theory. As we said, generation of power stroke and the cycle goes on repeating. So what happens if there is a depletion of ATP? There is no ATP. What will happen? The myosin head will remain attached to the actin, and then this state, which occurs after death, is known by the name of rigor mortis. It is a state of contracture because of the loss of ATP. It is one of the recognizable signs of death which are seen hours after the death, and all the muscle goes in a state of irreversible rigidity or contracture, and it becomes very difficult to manipulate a body. So on microscopy, if we see that there is a continuous interaction of actin myosin, they are in attached phase, no ATP is there because of the depleting sources of ATP after death, because organism cellular respiration does not occur. So the actin and myosin remain in the state of attachment. This we have already talked. So absence of ATP, no reuptake of calcium, then calcium level in sarcoplasm increases, continuous contraction resulting in abnormal, rigid and uninterrupted contraction. Then there is no ATP for relaxation and it remains in the state of bound to the actin filament. So the time is variable for rigor mortis. It may be 3 to 4 hours after death, maximum reaching in 12 hours, and then it disappears after 3 days. However, if the conditions are warm, then this process is accelerated, and in the end there is autolysis of the contractile protein, and then finally leading to the release of these actin myosin complexes because they are proteinaceous in nature. So the material for today's lecture was taken from these books, as you can see.